official title is In Vitro Min-Protein Patterns Arise from Self-Controlled Chaos. So the floor is yours.
Okay, so thank you very much. Today I'd like to present the work I'm doing with Fine on min-protein pattern formation and in particular pattern formation of reaction diffusion systems in general in context of quantitative mass-conserved reaction dynamics. And the goal of this talk is to show you that a spatial temporal order can emerge from spatial temporal chaos by controlling or reorganizing certain properties of the chaotic attractor. So we get a picture where we can get an ordered state from controlling a chaotic state. So chaos is not basically the end of order when you drive the system too hard or too far away from equilibrium, but it can be the origin from which you start to get organized patterns. So, and since we talk about reaction diffusion systems, we start with the work by Alan Turing. Alan Turing made a lot of important contributions, and one of these is his idea about the chemical basis of morphogenesis, and this idea goes as follows. So he considers a chemical reactor with some chemical species inside it, which is small, so in a well-mixed state, and it's in a stable chemical equilibrium. So let's say it's yellow, and if we take it and shake it, it remains yellow because it's stable. Now, he takes identical copies of these reactors and couples them diffusively. And what he finds is that if we have different diffusion coefficients for the chemical species inside the reactors, this can give rise to pattern formation only by diffusive coupling. So one can take this and describe this system classically as the exponential growth of Fourier modes, and one can easily compute the growth rates of each Fourier mode, and this is the dispersion relation, and the fastest growing mode then gives the length scale of the pattern that we observe at onset of pattern formation. So this is the classical, basically textbook picture that we have, and now we want to take this serious and apply these ideas to quantitative dynamics. So we assume we have real chemical interactions, so chemical reactions are mass conservative. So let's take the system in the pattern state and do the following. We put the walls back in, and then we ask the question, so what's inside these, again, decoupled reactors? And for simplicity, let's put some numbers on it. So let's say we have two chemical species, A and B, and in the original reactor we have 10 A's and 10 B's, and because of conservation of mass, we know that the total amount of molecules inside the whole pattern system. So the question is, what's inside these boxes? So how much of molecules do we have in these boxes? So let's first take the limiting cases. So the first limiting case is where the system returns to the initial uniform state. So there has been no net redistribution of mass, and for diffusion-driven stability, one can show that this is impossible intuitively, because having different diffusion coefficients means having different diffusive fluxes, so different exchange rates of mass. So this is impossible. The second limiting case is this one, where the system is basically frozen in the pattern state. So every single point in space, and remember we can make this partitioning as small as we like, every single point in space is in a stable equilibrium independent of the diffusive coupling to its neighbors. So this contradicts the assumption that we have diffusion-driven instability, and it would also imply that we can decompose every pattern and redistribute these individual reactors, build any kind of pattern with it, and it will always be stable. So these two cases can be rigorously ruled out. So the message here is that if we want to know what's inside these boxes, we have to actually look inside and count the numbers. And let's say we do find 5 A's and 16 B's in the blue one, and 15 A's and 4 B's in the red one. So we have mass translation, and for these numbers we can then compute the corresponding local equilibria, which are, let's say, green for the blue one and red, magenta for the red one. And of course, this is just a graphic way to say that they are different. The decoupled system returns to distinct equilibria, which are characterized by different local system parameters, speaking in the language of dynamic systems. So the picture of the two-ring instability is now that it takes a collection of identical reactors and it maps it through a pattern-forming process on a collection of non-identical reactors, which are characterized by different local parameters. So we can say that the diffusion instability creates a pattern in a local parameter space, and the characteristic length scale it sets is the length scale of parameter transport. And because we change parameters, the properties of the local systems can change. So the number of local equilibria and their stability can change, the type can change from oscillatory to non-oscillatory, and so on. So, of course, all of this was just a conceptual idea, and now we want to take this and apply this idea to a real quantitative system where we have the data, and the MinPris system in vitro presents itself as an ideal candidate. So we have just seen this in the talk by Simon, where we have two globally conserved protein species, MinD and MinE, and you put them in the simplified picture in a box. At the bottom you have a membrane attached. These proteins bind to the membrane, react with each other, and unbind. You have just seen the reaction scheme presented by Simon, and this gives rise to pattern formation, where you see spirals and traveling waves on the membrane. And the theoretical description is as follows. So we have an established model which reproduces all the patterns in vivo, in wild-type cells and in mutant cells. It just takes the experimentally verified molecular interactions into account and is formulated in terms of mass action law, and it takes the full system geometry into account. So the structure of the system is as shown here. So we have in the bulk, we have a three-dimensional reaction diffusion system. For the bulk species, we have a two-dimensional reaction diffusion system for the membrane species, and both these species are coupled through a reactive boundary conditions, which is nonlinear in this case. And now coming back to the local reactors, with which I started, we can now think of this structure as the local reactor, where we have this point here on the membrane, which is coupled to a one-dimensional diffusive line, and we will consider this as a local reactor, and we will restrict ourselves to this two-dimensional section of the three-dimensional geometry. So we will be looking at a one-dimensional section of the two-dimensional membrane. And the system height, the bulk height, serves in our simulations as a bifurcation parameter. There's a minimal bulk height at which Turing instability sets on, and we have tuned in the next slide the system directly above the onset of the Turing instability. The dispersion relation is shown here. So here you can see at the top the membrane profile after some long time has passed on the membrane along this one-dimensional line. You can see a very small amplitude pattern here. And now, in the spirit of these local reactors, we can take every single point here in space and count the local particle numbers. And here at the bottom, we will be plotting these one-dimensional numbers dynamically as the dynamics will evolve. So these are the blue dots here, and sigma and delta are just the rescaled coordinates of the global average of MinD and MinE. So sigma is just the sum, and delta is just the ratio of but don't let this confuse you. Just think of sigma and delta as some local counts. And because of global conservation, of course, the center of mass of this whole distribution will always remain fixed. And the a-axis here is the amplitude, so this is the local distance of a particular reactor from its local equilibrium. So if we now run the simulation, you see the system starts here in this white area, which is characterized by local stable fixed points as required by the Turing instability. And in this gray area here, the local reactors become oscillatory unstable. And the Turing instability spreads the system out in parameter space, which you can see here. And at some point, the system enters locally this unstable regime, and this is manifest on the membrane by the propagation of rogue waves, which propagate through the system due to periodicity. They collide with each other and annihilate each other, and the system then settles down in this state of spatial temporal chaos, which happens at onset of the Turing instability. And the corresponding distribution parameter space becomes very broad. Now, to emphasize these dynamics and what's happening, we will now focus on the dynamics of one single point here in space. So we pick this single point here, and here at this point in time, it is located here in this stable regime. So if we now isolate this single point, decouple it from the rest of the system, and run its own dynamics, it will return to a stable equilibrium, as expected. Now we couple back to the rest of the system and track its position, and what we find is that coupling to the rest of the system propagates this local point through its own local parameter space, and at some point in time it enters this unstable regime, and if we now decouple it from the rest of the system and run the simulation, we find that it now approaches the limit cycle. If we do this again, it again leaves the unstable regime, but re-enters it again at a different point in time, but at a different local parameter point. And if we now run the simulation, it again, of course, approaches the limit cycle, but a different one. And the same holds true, of course, for the stable equilibrium, which are always different. And of course, we can do this analysis for all points in space. So we can look at all these points in space, look at the trajectories through the local parameter spaces, and then look at the correlation of how the local stability of these local reactors change. And we compute the correlation of this stability exchange. And what we find is that in this state of spatial temporal chaos, there's no spatial correlation of the exchange of stability. So we can say that spatial temporal chaos here is characterized by a broad distribution in parameter space, local equilibria that are nowhere stationary, so they change all the time. And this exchange of local stability is spatially uncorrelated. Now this is the state of spatial temporal chaos. Now we drive the system further away from the Turing instability. So now we have a broader band of unstable modes. So this would be the regime where we will typically expect to see more disordered state because of mode coupling. But this is not what happens. Again, as before, we start in the stable regime. Again, the Turing instability spreads the system out in parameter space. Again, it at some point enters this unstable regime, and the amplitudes on the membrane become very large. But in contrast to the previous case, we see now some order emerging on the membrane. Here you see large amplitude standing waves and propagating wave packets. And in the distribution of local parameters, you see, in the distribution of local parameters, you see that the system approaches a narrow band in this parameter space. So if we look at the spatial correlation now, we see there's a finite correlation length of the exchange of local stability, and it decays oscillating with the characteristic length scale of this pattern. Now we increase the bulk height even further. So we have an even a larger band of unstable nodes here. And what we find here is there's a secondary local instability happening, so which will occur here, which is a target instability which sends out traveling waves here. And this band of traveling waves, it slowly invades the domain of standing waves. So we have transiently a chimera state where we have synchronized and desynchronized oscillator in one large system, and on a very slow time scale is then expands, so we can skip forward in time, as you can see here. If you look at the distribution of local parameters, we see that now the system starts to condense on a one-dimensional cycle here, which spans the stable and the unstable regime, and this is basically the attractor of the whole system, and these traveling waves here is what we also see in the experiment. So in the two-dimensional membrane, this would be the center of the spiral, the spiral pattern. If we now look at, so the red regions here are the points where the system is locally unstable. If we now look at the statistics of the distances of these red regimes, we find that the distance here is at onset quite precisely set by the Turing, by the fastest growing mode. So now behind this regime of spatial temporal chaos, this dispersion relation now enables us to compute and predict certain properties of the pattern as the length scale and also the speed with which these waves are traveling. So to sum up, the Turing instability in a mass-conserved quantitative system, we distribute a globally conserved parameter between the local reactors, so it takes a collection of identical reactors and it maps it on a collection of non-identical reactors with distinct local properties in terms of dynamical systems. We find spatial temporal chaos characterized by broad distribution in this parameter space, local equilibria that are nowhere stationary, and this exchange of local stability is spatially uncorrelated. The Turing instability now imposes control of these exchange of local parameters by this mechanism here, which creates a correlation between these properties of these local reactors and leads to a contraction of this distribution in parameter space to a one-dimensional line, which corresponds to the pattern which we also observe in experiments. So this mechanism is based on the self-organization of chaotic system, which we observe at onset, but not on the destabilization of a homogeneous uniform state. So with this, I would like to thank Irvin Freund and the group, and of course you for your attention. Thank you very much.
Student questions? As we're waiting.
Oh, that was truly excellent. So the business part of the mechanism is really the 2D part of the membrane, whereas the 3D external part doesn't seem like it has to be finely tuned. And isn't that related to the limit cycle existence theorems, which really only apply to 2D?
The 3D dynamics, even the dynamics above.
Yeah, above.
These are very important actually, but I haven't shown this to you. I can show it here. So there's a very complex pattern directly above the membrane. You can see it here. This is the snapshot of this propagating wave packet, and here you see this is the z-axis. So this is the membrane, this is the cytosol, and this is directly above the membrane, the concentration gradient. So this exchange of parameters, which is exchange of particles, happens in this layer, and there's a very complex structure emerging here in this layer.
But you could get the Turing waves without the 2D membrane component, I'm thinking, because again, limit cycle existence is dependent on 2D.
So
but
you don't need this third dimension. You can get similar dynamics, of course, in 2 dimensions as well. Yes. But the local limit cycles are unequal here. There's not these are not identical limit cycles. It's very hard to pin this down to a unique limit cycle, which you, like in the Kumamoto model, where you would, or complex Ginzburg-Landau equation, where you would think of the system as a collection of identical reactors or identical oscillators, which is not the case here. They're very dynamic.
Okay, great, thanks.
Further questions? Okay, if there's no questions, no further questions, let's thank Jacob again.